Hello, in this project my goal is to create a quantitative characterization of the landscape where Peromyscus polionotus is known to occur. This is a small nocturnal fossorial rodent endemic of the southeastern United States, particularly in Florida, Alabama, Georgia, South Carolina, and North Carolina. It is strongly associated with old fields, abandoned fields, and beach or dune habitats with loose, sandy soils, where it builds underground burrows and it tends to avoid the edge of its habitat. Its diet is mostly seed and invertebrates. Although it is globally listed as least concern, several coastal subspecies are of conservation concern. To describe the landscape where Oldfield Mouse actually occurs, I used the citizen science platform iNaturalist, and I filtered the records to include only research-grade observations. Under this criteria, I obtained 33 occurrences of Oldfield Mouse, all recorded between 2013 and 2025. To quantify land cover around this occurrence, I used 2023 crop data layer for the 5 southeastern states. Because this data layer does not fully cover coastal beach areas, I had to discard 9 of the 33 observations that fell outside its extent. These 9 points correspond to coastal form of Oldfield mouse, so the conclusion of this project applied mainly to inland populations. For the remaining records, I defined a buffer around each point based on the largest reported home range of the Oldfield mouse, about 3.2 hectares, which correspond to a radius of roughly 100 meters. With that buffer size, I used the Dissolve tool in ArcGIS Pro to merge overlapping areas and ended up with 20 independent buffers. From this set, I randomly selected 5 buffers that are sized where I will summarize the landscape metrics. To describe the landscape around this occurrence, I calculated a metrics in R with landscape metrics using the 4-cell rule because Oldfield mouse has small home ranges, relies on the core of patches, and tends to avoid edges, so diagonal connections are less meaningful for this species. At the class level, I focused on composition with percentage of landscape and on configuration with largest patch index and edge density. These metrics are relevant because they tell us how big the main patch of a class is and how much edge it has, which directly relates to whether Oldfield mouse is using large, compact core areas or landscape dominated by edge-heavy fragmented habitats. At the landscape level, I summarized composition using patch richness, Shannon diversity index, and Shannon's ifness index, and I described spatial connectivity with aggregation index and contagion. These metrics are relevant because they show how many habitat types are present, how evenly they are distributed, and how aggregated or fragmented the overall mosaic is. I initially considered core area metrics such as total core area, but they were not usable because many buffer classes were only 1 pixel wide, making core area essentially zero or undefined. Now I will show the result for landscape composition. Here I compared land cover composition at 2 scales. In the overall 5 states focal area, the landscape was dominated by evergreen forest, followed by woody woodlands and deciduous forest, with grassland and developed open spaces also contributing. There were 89 land cover types, but most of them were less than 3%. In contrast, within the 5 buffer, where Oldfield mouse was recorded, there was only 17 land cover types. The composition was also dominated by evergreen forests, together with shrubland, herbaceous wetlands, coron, and woody wetlands. At both scales, evergreen forest was the main cover type. However, we must remember that Oldfield mouse is associated with old fields and fallows rather than forests. Therefore, what we are seeing in the composition of the focal areas is that much of the species habitat occurs embedded within an evergreen forest matrix. Using the percentage of landscape in the 5 buffer and in the broader 5-state focal area, I built a habitat use versus availability plot that compares the proportion of each land cover class in the buffer with its proportion across the larger region. For habitat use, I also calculated standard deviation shows as error bars. Overall, most land cover classes were used in proportion to their availability, so there was not selection for many land covers. A exception was evergreen forest. The observed use of evergreen forest was not consistent with the ecology of Oldfield Mouse, as mentioned before. It should be reflecting evergreen-dominated matrix rather than true use of forest cover. Negative selection was detected for grassland and other hay non-alfalfa. This apparent avoidance was ecologically plausible, but it may also have been reinforced by land cover classification issues and sampling bias. Woody wetlands and deciduous forests appear to be underrepresented, but the large standard deviation and a small sample size likely biased these estimates. Nonetheless, they could represent another land cover matrix. An important limitation was that I used the 2023 crop data layer while the iNaturalist observation spanned multiple years before and after 2023, so the distinction between active crops and fallow or oilfield was not always reliable in time. Regarding the landscape metric percentage of landscape within the buffers, on average, the five buffers were strongly influenced by evergreen forests, with shrubland, herbaceous wetlands, cotton, and woody wetlands contributing roughly from 10 to 15%, while all other covers were secondary or rare. The most relevant potential habitat for oldfield mouse was the combination of shrubland, fallow, peanuts, and cotton. For the class level, largest patch index within the buffers, evergreen forests, shrubland, herbaceous wetlands, and cotton had relatively high mean values showing that in some buffers a single continuous patch of these covers occupied most of the landscape. In contrast, grassland pasture, fallow, and developed classes had consistently low largest patch index values, indicating smaller and more fragmented patches. This means that apparently for the oldfield mouse what mattered was not only how much potential habitat was present, but whether it formed large continuous patches. Cases where cotton, peanuts, or shrubland were the largest patch suggested buffers with extensive open or semi-open habitat that could be favorable. For the class level, edge density within the buffers, evergreen forests, woody wetlands, and shrubland had the highest mean edge density values and very large standard deviation. This indicates that, across buffers, these covers were sometimes absent and sometimes formed very dissected patches with a lot of boundary against other land cover types. For the oldfield mouse, high edge density in forests and wetlands means many sharp transitions between open habitat and dense or wet cover, which likely increased edge exposure and movement goals for a species that tends to avoid edges. In contrast, agricultural and open covers such as cotton, peanuts, fallow, and grassland pasture generally showed more moderate edge densities, suggesting more compact open patches where oilfield mouse could move up forage with less contact with unfavorable edges. Here I showed the landscape level metrics for composition and connectivity within the buffers. Patch richness ranged from 4 to 8 classes and Shannon's diversity and ifness indicated moderate diversity but generally uneven landscapes, where one or few land covers tended to dominate. For all field mouse, this means that occurrences were not in extremely simple or hyperfragmented mosaics, but in landscape with a few to several cover types, usually with dominant class. Aggregation index and contagion both pointed to intermediate levels of clamping and mosaic structure. Some buffers were more aggregated and dominated by one cover type, while others were more mixed and interspread. To summarize, landscape used by Peromyscus polionotus show moderate richness and are typically dominated by one or few cover types. The species is associated with fruit and annual agriculture. The configuration metrics indicate that several buffers contain large continued patches, and the connectivity metrics show intermediate aggregation and contagion.